purification of plasmid DNA. We all know that many bacterial species have, other than the genomic DNA, small plasmid DNA, which are also closed circular DNA. And there have been a lot of protocols that have been standardized for purification of plasmid DNA, as plasmid DNA is used as a cloning vector in recombinant DNA technology. So let us look at the learning outcomes of this session. In the protocols for plasmid DNA isolation, the same general strategy is used for genomic DNA is used for the genomic DNA isolation. That means that from the cell extract, which contains all the different biocomponents, one by one certain components are eliminated, so that at the end what remains is the molecule of interest. Now, one major aspect in this case is to make a distinction between the plasmid DNA and the total cell DNA. And this is possible due to differences in the size and conformation of the two. There have been several protocols developed for plasmid DNA isolation, but the most prevalent one, and that is used in the labs around the world, is what is called as the alkaline denaturation method. Ethidium bromide cesium chloride density gradient centrifugation can also be used to separate plasmid DNA from the genomic DNA. Now let us look at the workflow of the isolation of plasmid DNA that, as we suggested, is very similar to that of the genomic DNA isolation. So first, one obviously has to get a good biomass. So therefore, growth of bacterial cells in an appropriate medium is what is the initiation or initial step of the isolation of the plasmid DNA. Thereafter, the cells are harvested, and these harvested cells are resuspended in a buffer. And then, having resuspended the cells, these cells are subjected to cell lysis, but in such a way that the genomic DNA integrity is maintained. Here, what is important is that the DNA, that is the chromosomal DNA, should not get denatured or should not get fragmented into small fragments. Once the cell lysis has been done, then the next step is to separate the plasmid DNA from the genomic DNA, because the cell extract obtained will have both the chromosomal DNA as well as the plasmid DNA. Once the plasmid DNA and the genomic DNA have been separated, thereafter protein and RNA can be eliminated. And once protein and RNA is eliminated, what is obtained in the aqueous layer, or what will be obtained due to elimination of all the other components, is the plasmid DNA, and this plasmid DNA can then be precipitated. The precipitated DNA is resuspended in an appropriate buffer or in distilled water, and the quality and the quantity of the isolated plasmid DNA is assessed at 260 by 280 nanometers. Now, this plasmid DNA that has been obtained may be modified in the lab for its use as a cloning vector. Now, the separation of plasmid DNA from genomic DNA is basically possible because there is a difference in the size of the plasmid DNA and the genomic DNA, with the plasmid DNA being much smaller compared to the genomic DNA, and of course in the conformation of the two. Now, let us first look at the separation of the plasmid DNA on the basis of just size. So where you have a bacteria which has a genomic DNA which is much bulkier, as you can see, than the small plasmids, and you can see that within the bacterial cell there can be several copies of the plasmid DNA present. And also what is interesting to note is that in many cases you may have more than one type of plasmid DNA present in one bacterial cell. There are a lot of attachment points where you have the chromosomal DNA or the genomic DNA also attached to the cell wall. So when separation has to be done on the basis of size, care has to be taken that the genomic DNA is more intact or more or less intact, as fragmentation of the linear genomic DNA would lead to linear DNA, but smaller fragments also may be possible. And if they are smaller fragments and the fragment size is comparable to that of the plasmid DNA size, then one will not be able to separate the two. So therefore, one has to ensure that the chromosomal DNA is present as a much bulkier or as a much longer fragment or a much larger DNA compared to the small plasmid DNA. So the first step basically is to um, have a controlled cell lysis, or rather cell wall removal mechanism. So when one says controlled cell wall mechanism, the idea is to ensure that the cell does not disrupt during the cell wall degradation or the cell wall removal. So the controlled cell lysis is carried out in presence of lysozyme. So we all know that lysozyme is able to break the glycosidic linkage between N-acetylglucosamine and N-acetylmuraminic acid in the peptidoglycan layer, and therefore the cell wall is going to get be degraded. Moreover, when EDTA is used, EDTA chelates magnesium ion, and magnesium ion is known to stabilize the cell wall. So when magnesium ions is removed or metal ions are removed, then the EDTA is able to weaken the integrity of the cell wall, and therefore the two together is able to take care of the cell wall present around the membrane of the bacteria. A 25% sucrose solution is added in this place to maintain the isoosmolarity, for the simple reason that when the cell wall is broken and the cell membrane is exposed, the cell membrane uh, should not be facing either a hypertonic or a hypotonic condition, because if it faces a hypertonic or hypotonic condition, then it is possible that the cell will burst open, and that is something that needs to be prevented. Why? Because if the cell membrane gets disrupted, then there is a possibility that the genomic DNA which is attached at points to the cell membrane can also get damaged and broken down into smaller pieces. So you would definitely get fragments of DNA which are linear, but what is taken care of by adding 25% sucrose is that the cell membrane does not lyse suddenly or does not disrupts, taking care of the fact that the integrity of the chromosomal DNA is maintained. What you achieve, therefore, by this controlled cell lysis is something that is called as spheroplasts. So, spheroplasts are cells which have remnants of cell wall present on its membrane, and that is exactly what is happening because of this treatment. Thereafter, the cells that are now spheroplasts are subjected to further lysis by Triton X100. Triton X100 is a mild detergent, and it is enough to remove the lipids from the bilipid layer of the membrane, and thereby it makes the membrane leaky, and eventually what is going to happen is that the cell is going to lyse to form a cell extract. So the cell extract will contain all the components of the bacteria, and in the components of the bacteria would be the linear fragmented DNA. To a certain extent, linear DNA may not be fragmented as well, and of course, the small plasmid DNA will be present along with proteins, RNA, carbohydrates, etc. Now, this is, or the cell extract is then taken in a centrifuge tube and centrifuged with the objective that on centrifugation, you will have all the cell debris, that the cell wall remnants, etc., getting pelleted out as cell debris, and the larger DNA, which is part of the genomic DNA, will form a layer just below, close to the cell debris, while the lysate or the supernatant would have the small plasmid DNA. Because they are small in size, they do not settle down, they remain in the supernatant. Now, this definitely is an easy method, But there are many times where one cannot guarantee that there is no linear DNA or the genomic DNA present in the supernatant if you have, due to the process, small DNA fragments being formed. And if small DNA fragments are formed from the genomic DNA, then they would also be present in the uh, supernatant. So you will not get a pure plasmid you'll not get a supernatant with just plasmid. You will also have small linear DNA fragments. The other possibility is that if the size of the plasmid is large enough as a fragment of DNA, then it is possible that the plasmid too is going to settle down, and therefore you would have hardly any plasmid in the lysate, but you will have more of the plasmid present in the pellet. And now, therefore, you're coming back to square one where the genomic DNA and the plasmid DNA are present together. Our entire objective is to ensure that one is able to separate the plasmid DNA from the genomic DNA. Now let us look at the separation of the plasmid DNA on the basis of conformation. So we all know that plasmid DNA, which is a closed circular DNA, in this form you know that it is a relaxed form. There is no supercoiling observed in this closed circular DNA, and also the supercoiling phenomenon itself is dependent on the fact that the DNA has no free ends. And so since the plasmid has no free ends, you can have the plasmid DNA undergoing supercoiling. Now, suppose one of the strands of the closed circular DNA is nicked, then it is possible that you have the open circular form present. And in this case, therefore, what is going to happen is that the plasmid is going to be always relaxed. It cannot supercoil because you have a nick which is making one of the strands at least free at its end. So, therefore, supercoiling will not be possible or will be minimized. And of course, one has the supercoiled DNA itself, then the supercoiled DNA is much more compact in comparison to the two. So the movement also, therefore, you can see how the plasmid DNA conformation can vary depending on whether it is highly supercoiled or it is not supercoiled at all. For plasmid DNA, what is most prevalent as a conformation is the closed circular DNA. Now, therefore, uh, when you have the understanding that you have to separate out the genomic DNA from the plasmid DNA, the supercoiled DNA and the non-supercoiled DNA can be distinguished. Why? Because when we are looking at the first step where cell lysis occurs and the cell membrane at certain points that attach to the um, chromosomal DNA, then due to cell lysis, it is definitely possible that the chromosomal DNA is also fragmented, larger fragments, but it is fragmented, and so it becomes linear. So therefore, the DNA, the linear DNA or the chromosomal DNA is going to be non-supercoiled, whereas the plasmid DNA is going to be supercoiled. And it is possible to separate out supercoiled DNA from linear DNA. Now let us look at one very prevalent method or one very common method that is used across different labs in different parts of the world, and that is what is called as the alkaline denaturation method. As the word suggests, you have the DNA getting denatured thanks to alkaline conditions. So therefore, understandably, the cell extract that is obtained after cell lysis is treated with an alkaline buffer, with the range of the alkali being anywhere from 12 to 12.5. And in this narrow alkaline pH range, any linear DNA fragment will get denatured, but a supercoiled DNA will not get denatured that easily. So, this basis of the fact that non-supercoiled DNA uh, can get denatured while the supercoiled DNA does not get denatured is being made use of to isolate the plasmid DNA or to separate the plasmid DNA from the genomic DNA. So as suggested, non-supercoiled DNA denatures while the supercoiled DNA does not denature. So here is the pictorial representation. So these are the linear DNA and these are the supercoiled plasmids. So when you treat this to pH 12 to 12.5, you can see how the linear DNA undergo denaturation to get single-stranded DNA. And when this extract, once it is treated with the alkaline buffer and it gets denatured, that is the non-supercoiled DNA get denatured, the extract is again neutralized by using an acid or a mild acid um, or an acidic solution so that the pH comes back to 7. Now when that happens, this pH is conducible for the single-stranded DNA to again interact with each other. And therefore, in the possibility of forming the double strand again, you will find that many of the single strand DNA get tangled to form a mass or to form an aggregate. So this tangled mass of linear DNA will get centrifuged out, but the supercoiled DNA will remain in the supernatant. So again, you are basically finding that all the genomic DNA, that is the chromosomal DNA, should be present in the pellet, while the supercoiled plasmid should be present in the supernatant. So here we said that acid has to be used to neutralize the pH, and so here what has been observed is many protocols use sodium acetate to neutralize, and this sodium acetate can also denature proteins and RNA. So therefore it is one target but with multiple facets. So the supernatant as observed over here will contain the plasmid DNA, and therefore this separation that is happening is not just based on size but it is also based on conformation. Another method that is used to separate the plasmid DNA from the genomic DNA is ethidium bromide CsCl chloride density gradient. Now we all know that ETBr is a ringed structure that can intercalate between the base pairs of a DNA. So when it gets intercalated, what happens is that the DNA becomes less wound or it begins unwinding. So essentially what we are looking at is that when there is no ETBR in the DNA is comparatively more denser than a DNA which is intercalated with ethidium bromide. So to take advantage of the fact that ETBR present in the DNA is changing its Bowen density if it is a linear DNA, then in a ETBR CsCl density gradient centrifugation, we all know that depending on the buoyant density of the molecule will get separated out. So here is what you're looking at is these are the CsCl density gradient centrifuges, and the density of the CsCl is such that the proteins do not even enter. So it is much less denser than the CsCl, while RNA is more denser than the CsCl that is used. And if the DNA is also loaded on this and is centrifuged, it has been observed that the linear or the open circular DNA will form a different band which is at a lower density compared to the supercoiled DNA which is present at a slightly higher density. So when ETBR is getting intercalated within the linear and the open circular DNA and also supercoiled DNA being highly compact will take up very few ethidium bromide molecules, whereas the linear and the open circular DNA which is relaxed can be able to take up many ethidium bromide and intercalate it within the structure, and that is definitely going to make the linear DNA or the non-supercoiled DNA more you know unwound, and when it is more unwound it becomes less denser. So therefore, the bond density of a linear DNA is going to decrease as much as 0.125 gram per centimeter cube, while in case of the supercoiled molecule, because it is not taking up too many ethidium bromide molecules, its density is only changing by about 0.085 grams per centimeter cube. So you can see how the plasmid DNA or the supercoiled DNA from 1.7 grams per centimeter cube does not move much up, while the linear DNA or the open circular DNA will definitely have lower density than the supercoiled DNA itself, and you can separate out the two thereby. When one wants to remove only the supercoiled plasmid DNA, one can take it out specifically with a syringe. But then what is understood is that this DNA has within it the intercalated ethidium bromide. And if you want to use this plasmid DNA for cloning, then that ethidium bromide has to be removed from the structure of the plasmid DNA. So the supercoiled DNA is collected with a syringe and then it is subjected to treatment with N-butanol. And when it is subjected to treatment with N-butanol, the ethidium bromide enters or gets extracted into the N-butanol while the DNA remains in the aqueous layer. So on centrifugation, one gets two distinct phases. The organic phase of the N-butanol will contain ETBR, while the aqueous phase will contain the plasmid DNA. Now, what further is also observed is that this DNA would also have cesium chloride present, as it was present in cesium chloride over here. So to remove the cesium chloride, the DNA is taken in a dialysis tube and is placed in a buffer. So when it is placed in a buffer, cesium chloride being a salt will move out to the buffer uh, because of simple diffusion. And therefore, what is going to be left behind is a concentrated plasmid DNA solution, which is going to be very pure or quite pure. And if it is not pure, then one knows then what can either do phenol chloroform treatment to remove the RNA and to remove the protein, or one can go in for sodium acetate as well to ensure that protein is removed from that. So therefore, when one wants a high yield of plasmid DNA, then it is definitely imperative that if the number of plasmids that are present within bacteria is high, then one would definitely get more amount of plasmid DNA. So therefore, there are strategies to improve the copy number of the plasmid. So you can see this is a bacterial cell with the chromosomal DNA and several plasmid DNA. And these are multi-copy plasmids, but one can increase the copy number further by incubating the bacterial cell with chlorophyllocol. Chloramphenicol, as we know, is something that is going to stop protein synthesis. Now, the amount of chloramphenicol that is able to kind of bind to the ribosome and lower down the protein synthesis is definitely going to affect the genomic DNA replication, the cell division, etc. But what has been observed is that uh, it it is able to allow the plasmids to multiply, and therefore you would have the copy number of the plasmid increasing, but otherwise the bacteria is more or less standstill. So inhibition of protein synthesis affects the chromosomal DNA replication, but it does not and cell division, but it does not affect the plasmid DNA replication. So the plasmid will multiply. Increasing the plasmid copy number therefore leads to an increase in the yield of the plasmid DNA. So let us make the conclusions. Plasmid DNA isolation is based on the same principles as used for isolating genomic DNA. Plasmid DNA can be distinguished from genomic DNA based on size and conformation, which becomes the basis of separating the two. Alkaline denaturation method involves treating the cell extract with alkali such that the pH is between 12 to 12.5. And this leads to relatively much greater denaturation of linear DNA than supercoiled closed plasmid DNA. ETBR CsCl density gradient centrifugation can separate supercoiled plasmid DNA from non-supercoiled linear chromosomal DNA. Therefore, protocols for plasmid DNA isolation has been developed so as to get pure plasmid DNA for its use in recombinant DNA technology as an efficient vector. Thank you.